So I only have half an hour, and I like to gab. So I'm going to start so that we try to get as much stuff in as we can in this 30 minutes. First off, I just want to introduce myself, Thor Rollins. I've worked at Nelson Laboratories now for almost 20 years. And on all that time has been in biocompatibility. I'm now the director of our advisory service group. Basically, we just try to solve problems, right? So, and I'm also a participating member of the TC 194 working group, which is the working group that writes the ISO 10993 standards. So first off, sorry, we've kind of messed things up recently, trying to make sense of it right now for you. I do have some things up here if you want to grab some afterwards. Probably the one that goes the fastest is our slide rules, for those of you who have not seen them, right? So this is the ISO 10993 chart in what engineers love to play with, which is a slide rule. So if you want to grab one afterwards, please do. If we run out, we have more at our booth, which is 3125. 35. Oh, I failed. 3135. So you can grab some more at our booth if afterwards. Also, a lot of times people ask me for a copy of my presentations. I'm happy to give them out. All I'll need from you is a business card. If you just want to drop it here or something like that afterwards, I'll make sure you get a copy of the presentation, okay? So let's go ahead and jump into it. It's been such a crazy couple of years. I know we've all been through this pandemic. This is my first time talking to a group of people in face-to-face in like 2 years. I'm so excited to see people. Hopefully you guys like this too, and you're sick and tired of webinars like I am and giving them. So I'm super excited to actually do this in front of people. So hopefully I'm not too rusty. But there's been a lot of changes in biocompatibility, specifically when it comes to regulations and how the FDA and notified bodies have started to look at things. I mean, the MDR came out during the pandemic. The FDA has changed. So we're going to try to cover as much as we can in 30 minutes. This is going to be kind of just high level, going through the most important things on these topics. And then obviously if you guys have questions about a specific one, we can answer those, or we'll back to my booth that's 3135. Yes, and we can talk there a little bit more too. So to kind of give you a sense of the topics that I thought were the most important to kind of high level talk about today during the presentation, we're going to talk a little bit about the force behind these recent changes. Okay, why are we seeing these changes in the industry? We're going to talk very high level on extractables and leachables. And the reason why I say this is because we could literally do 4 hours of E&L discussion, and I have, right? So if you have specific questions about extractables and leachables, please come back to my booth. We'll sit down, we'll break out some waters, and we'll spend the afternoon talking about it. But in this presentation, I'm only going to be able to hit on the top things there. We're also going to talk about an exciting new standard, 10993-23, which is a new standard, right? It's the new in vitro irritation. So we're going to talk about what that standard is, how is it being applied and accepted around the world right now. We're also going to talk about the pre-submission process with Presubs and what we're seeing right now versus what we saw pre-pandemic. And then last but definitely not least is during the pandemic, the FDA released a new draft document for skin contacting materials. And they talked about how we can kind of maybe look out getting out of testing for skin contacting materials. So we'll talk about that draft and where we currently use it today versus where we can't use it. So this is the agenda, and we're going to try to cover as much of this as possible in 30 minutes, okay? So stay with me. So why are we seeing these changes? Now, before the pandemic, the 10993 standard -1 was released. And -1 was updated to try to be a risk-based approach. If you've listened to some of my webinars, you know I talk about this all the time. I love this approach. The concept is that everybody uses basically the same materials out there, right? Now, right now there's kind of a shortage of some of those materials, but for the most part, everybody uses the same materials. So why are we continually testing the same materials over and over and over again? Most of the reason is because of processing. We tend to leave stuff behind when we manufacture these materials. But we should be able to try to understand more about the process when we do these materials and the materials themselves to justify out of testing. I can't tell you how many times in my 20 years I've tested stainless steel. I mean, it's ridiculous. I can tell you for certain stainless steel is safe because I've put it through so many different types of tests in 20 years, it's ridiculous, okay? It's not stainless steel I'm worried about. It's what you're putting on stainless steel afterwards, right? The cleaners, the detergents, oils, things like that. So we can just follow the 10993, but making the 10993 more risk-based has also made it harder to follow. It's no longer a checkbox, right? So we're going to go through that. This is the old FDA guidance, or the old FDA document, right? Guidance document. G95 is what it was called because it came out in 1995. I'm old, and that's the year I graduated high school, right? So this was extremely outdated. So it was great that the FDA in 2016 released a new guidance document, but that this old document was this checkbox approach. And everybody did it this way, right? So you looked at what your testing, your device was, how it was used, and you just did the testing. It was easy. And I joke, and it's not a joke because I'm this nerdy, but I actually taught my daughters how to do this. They literally could categorize a device. They could work for a lot of you guys. So it was so easy that a 10-year-old daughter could do it, right? That was the checkbox mentality. The problem with that is we did not have to know anything about the device, the materials we're using, or the processing. As long as it didn't fail a sensitization test or an irritation test, we felt great. We felt like it was, safe. Then we started realizing how inappropriate sometimes these animal tests, how they're not as predictive as we thought they were. And we started seeing problems in the industry where these complicated materials like nanomaterials or combination devices, things like this, that these tests were not really designed to screen. So we came up with a risk-based approach. And this is the new 10993 chart that's in currently. And as you can see, we added a new column, which is physical or chemical information. So this is required. This does not require chemistry. I've had tons of people, notified bodies, tell me that this says that you have to do chemistry. It does not. It says you have to do physical and or chemical information. So you have to gather information about your materials and your processes and understand the risk. And then based on that, then you can evaluate the testing needed. It does not mean you have to do chemistry. It means chemistry fills in the gap if you don't have understanding. So that's an important thing to remember, and that is something now we're starting to be able to get notified bodies specifically to understand. FDA also understands this, so this is a part of their new guidance document, which basically talks about risk assessment. It talks about understanding the materials and the processes, like we've talked about, and then the risks from that, from a biocompatibility perspective, and then you look at a plan, I love that plan part there, should be developed to mitigate those risks. So this is the whole biocompatibility concept from the FDA. Starts with the biological evaluation plan. Those are so important right now. If you have anything that's a little bit weird, and I mean something you want to justify or something you're not sure about, anybody should develop a biological evaluation plan. Obviously, we can help with that, but anybody can do it if they know what they're doing, okay? Okay, so now that we know we should develop a plan and we know what kind of risk assessment we need to do, let's look at the current trends in extractable leachables. For those of you that may not be familiar with extractable leachables, sometimes people call it material characterization, sometimes they call it chemical characterization. It really is the same kind of thing, which is we take your device, we put it into a bunch of fluids, we extract it, and then we identify and quantify the chemistry coming off of it. This is great because the old animal tests, we would just inject extracts into these animals, stand back and say, whew, nothing happened, but we had no idea about the potential risks that device really had. With chemistry, we get to actually see the chemical fingerprint of your device, and then a toxicologist can make an assessment. The problem with chemistry and why it's so frustrating for so many of us, it's not standardized, right? So the old animal tests, pretty much every single lab did the exact same tests, exactly the same. There was no deviations as far as, there were slight ones, but really they made no difference. With chemistry, there is no standardization. Each lab's gonna do it different. Honestly, each lab's there's gonna be different variations on how good or how bad that chemistry is. I can tell you right now, depending on the risk of your device, depends on how good your chemistry needs to be, right? So if you just have like a sling, you could probably get by with some pretty bad chemistry. The risk isn't that good. But if you have a heart valve, you better have the best chemistry that you can get because the risk of that heart valve is so much more, right? So coming from a lab that I think we have pretty good chemistry, I'd love to tell you, you need the best chemistry every single time, but that's just not the case. If it's a low-risk device, don't do crazy chemistry. Do the chemistry necessary for the risk of the medical device, okay? But if you have a high-risk device, make sure you're doing the best chemistry possible. But with this, we have much more emphasis on this risk-based approach because you have to design your test protocol up front. You just can't give it to a lab and say, okay, do your chemistry. You should be working with your lab or an expert to develop the right chemistry for your device. That's new because before, you just submitted devices to a lab and said, okay, do the testing. Now you have to work with the lab or work with somebody to be able to help generate the appropriate testing. Also, we have to look about cumulative exposures. So chemistry changes if a device is used every day versus once, right? So even an implant, the way I like to explain this is if you have a permanent implant, a lot of people think that's a high-risk device. But chemically, it might not be. Because if it's a piece of stainless steel, all that chemistry is going to come off immediately, and then over time, you're going to have almost no chemistry come off of it. But if you give something like an IV bag, that a patient sees a new IV bag every day they're in the hospital, the new dose of chemistry comes every day. That's much more worse from a toxicological standpoint than a permanent piece of metal in your body, okay? So we have to evaluate that based on the chemistry, where before we really didn't have to worry about it too much. Also, we have a combined materials. We have materials that don't work well with certain extracts. It's a very complicated process. It's a great process that gives you good information, only if it's designed correctly. That's the important part. So please get comfortable with somebody who can help to design these chemistry tests because they become important. They can really mess you up if they're not done right. I've seen it millions of times. So also, with notified bodies, the problem we're seeing too with the new MDR, they're starting to require chemistry for the new 10993-1. So we have all these medical devices that have been on the market for 20 years, and they did their traditional biocomp tests, if they did any biocompatibility whatsoever, and now they have to submit to the new MDR, and now they're asked to do chemistry. Now we're starting to see things come off these medical devices that we didn't think were there, or maybe didn't realize they were there. We're starting to run into some issues. You'll see some recalls and things like that based on this type of information. So it's one of those things where maybe ignorance was bliss, but from a patient standpoint, I'm glad we're starting to understand more about what chemistry leaches off these medical devices. So the other thing I wanted to touch on is we're having an interesting impact right now with Brexit. So, and this involves with E&L. So Brexit's moving away from the EU. So right now they're still accepting the CE marks. But soon the MHRA is going to have their own mark that you have to apply for. They have their own anticipation on how to do chemistry and what expertise is needed for that chemistry. So there might come a time where you don't have to just worry about Japan, China, FDA, notified body, but also MHRA. And they're going to have different recommendations than everybody else. So we do have to kind of understand what Brexit is doing to the medical device field too. Also, we've learned a lot from the MDRs now. We've learned, like I said, about how to do correct chemistry for the risk of the device. Please don't do overboard chemistry like I talked about. We also learned about how to communicate with the notified bodies up front to really understand, because they're learning about the MDRs too. If you want to kind of understand what others are doing to save money, let me know. We've worked with a lot of clients on family groupings, on materials evaluations, to really reduce the testing needed for MDR submissions. So that's something that we've been very successful in recently. Okay, one of my favorite things to talk about is this new 10993-23, okay? So the new 23 was introduced just recently this year. And for those of you who don't know, we took 10993-10, which is sensitization and irritation, and we broke them apart. So we kept sensitization in -10, and we took irritation into -23. The other thing that's very important there is we actually included for the first time an in vitro version of the irritation test. This is the test historically we've always done in rabbits. 3 rabbits, injecting 5 times in each rabbit. We did some math to come up with an irritation score. Now we can do this test in vitro. That's great from a cost standpoint. It's actually a little cheaper, it's quicker, and it doesn't involve animals. Okay, so let's go through that real fast. So for those of you that don't know what irritation is, it's a little quick personal example that helps put it home. It's embarrassing, but I think you guys are okay, right? So really fast, my wife went away with some friends on a night out, to spend some time with friends and have a girls' night out. I have 3 daughters, so I thought I'm a dad, I can parent, right? So this isn't a big deal. I decided to do laundry to try to help out, while my wife was having fun. We were out of laundry detergent, so I went to the store. The laundry detergent that my wife uses was right in front of me, and it was kind of expensive. But on the very bottom drawer there, the bottom shelf, there's this like non-brand stuff that I thought, it's just soap, right? It's not gonna make any difference. So I bought that, brought it home, did a load of laundry, and that just happened to be pajamas for my girls. And so I ended up putting them in their pajamas, put them into bed, thinking I was gonna be husband of the year. My wife got home the next day, my girls woke up, guess what they had on their body? Yeah, a rash, right? They had an irritation reaction to the detergent. This is what we look for in the irritation. And honestly, most of the time it's either soap related that we see a positive or some kind of chemical residual that's an irritant. So and usually this is how we look for it on rabbits, right? We look for that same response that my daughters had, which is that redness and swelling. As you can also see, I want to point home, the category right here for irritation is an E. It means you do not have to do irritation if you don't think it's necessary. You should evaluate the need to do irritation, okay? So make sure you remember that. So it always means evaluate. Okay, so historically, we did a non-polar extract and a polar extract, and we inject them in bunnies. And we look for that irritation response that my children had. So I know my embarrassing situation, but now you remember forever, right? That's what an irritation response is. So we'll look for that in rabbits. Now we've replaced the rabbits with reconstructed human epidermis. It's actually human skin from stem cells that we've grown up into little dishes. These skins are almost exactly like your skin. They mimic it pretty much exactly. They're actually a little looser. They miss some of the connective tissue, so they're more sensitive than our skin. But the concept is the same. And the ISO says you should consider the in vitro test first. So before you jump to an animal test, you should consider the in vitro first, okay? So these are the general principles, the steps you should go through now for irritation. You should look at the literature to see if your chemistry has irritants in it. If there's something on your device you're using that's an irritant. So you gather physical or chemical information. Then, if you can't justify out of irritation at that point, you do the in vitro irritation, okay? Then, if you needed, so if something happened in the in vitro, or if you're in a category that can't use the in vitro, then only then do you go to the animal tests. If the animal test fails, you have to go to some kind of clinical evaluation to truly understand the risk from an irritation, okay? This is the procedure that 10993-23 outlines. There is a little bit of a difference here. If your device has one of these special tissues, okay, then they just recommend going directly to the animal test. We're looking to change this. We are getting some of these in vitro tissues made up. But right now, the validation was only done for the skin contacting. So if you do, vaginal, penile, ocular, we still have to do the animal test of those right now. Eventually, we're hoping to change that. But this is the difference. So this is our skin, right? And this is the reconstructed human epidermis. Very similar. There's a little bit of difference, but really we're able to look at the exact same response in both. Okay, so I'm going to walk through very high how we run this test, since this might be new to some of you. It's actually exactly like the animal test in these 4 different areas. So the first one's extraction. We follow the exact same extraction procedure as the animal test. We extract at 37 degrees for 72 hours or 50 degrees 72 hours based on the contact time of your device. We also still extract in saline and oil. So exactly like the animal test up to this point. The exposure is a little bit different. So Typically, we apply them to these tissues. So you can see we do a little bit, 100 microliters of this up to 3 tissues, so we run them in triplicate. So instead of injecting it into the rabbit, we actually put a little bit of this extract onto these tissues. Then what we look for is cell viability. We look at it using an MTT test. An MTT test looks for healthy cells. So healthy cells, non-irritant cells, will turn purple. So we want to see purple. So as you can see up here, the top layer is pretty purple. That means no irritation. Then we start seeing these 2 on the left. Those are pretty clear, and the ones below that has just a little bit purple in it. That means the ones on the left, those 2, are actually our positive control. And then the ones below that are starting to form irritation. So we are able to run these on spectrophotometers for purple and measure how much purple's there. The more purple, the less irritation. The less purple, the more irritation. Pretty simple, right? You can see why this is easier to run, cheaper to run, and better to run than the animal tests. Last is how we analyze the data, how we look at the data. So right here, you can see that these are how much purple. So those 3 columns on the right have lots of purple. The saline with a little bit of irritant on there has a little bit less, but the positive control is completely gone. So that shows us that we're having cell death or an irritation response. We can confirm this type of irritation response with an interleukin test. So if you have an irritation response, the cells release interleukins, and we can look for those to confirm it. But most of the time, honestly, I just want to tell you, the irritation test right now in the industry fails less than 1% of the time. The sensitization test fails less than 0.01% of the time. So we're running on all these animals every day for less than 1% and less than 0.01%, okay? So if we can find valid irritation or in vitro alternatives, it's better for everybody. It's not that high of a risk, and it's better for the animals and cheaper and quicker for the consumers. So I do want to just say right here, we can do the irritation on all devices, gels, liquids, solid devices, it doesn't matter. I do want to mention right now that every single regulatory body in the world accepts -23 except the FDA, okay? I'm sorry, I know that was but like every notified body, Japan, China, Canada, everybody accepts this, except for the FDA. We're working on that. So we've been working with Medtronic on something called an MDDT, which is like a investigation kind of thing for new procedures. We're convincing the FDA that they're the only people holding up the use of this test. Because if the FDA doesn't accept it, no one's going to use this test, right? Let's just be honest. That's not true. Some European customers who only do in Europe use this test, but the vast majority of you out there are going to be submitting to the FDA. So we're close. We just got a meeting with them set up soon to go through the final results that we have for them to prove that this test works well. And hopefully by the next couple months, we have the FDA on the bandwagon and we can start running this test like it's supposed to be in the standard. The FDA is feeling a lot of pressure because it's in a standard, it's a standard method, and all the other world's accepting it, right? So it doesn't look great for the FDA right now. So this just kind of goes more into how to classify if it's an irritant or not, the acceptance criteria. Like I said, if you give me your business cards after this, I'll give you the deck so you can actually have this to reference. Okay, so let's go to, and by the way, if you guys do have questions, the best way to shut me up is raise your hand. If not, I just keep on going, okay? So if you have questions, go ahead and raise your hand on any of these. The next one is the pre-submission process. I've been kind of heartbroken over this because when the pre-submission process came out, I was a huge fan of it. The possibility of meeting with the FDA for free and talking to them sounded like the best thing ever, okay? And so that process is this. So this is the actual document you fill out, right? So here is the pre-market review submission sheet and the request for feedback. So the concept was awesome. It's informal. They will say it's non-binding, but most of the time if they say something here, I've held them to it and they've been good. You go in, you're able to talk to them about test methods, about procedures, chemistry, all these things, and it's been great. The concept was great, and at first it was working out really well. So they used it, the FDA loves it. They refer to it as a QSub, it's a Presup, the Q identification number. And we've done this a ton. We've helped clients submit for the Presup. We've helped them run the Presup. We've sat on the Presup to support them. It's something we do almost every day. There's a lot of benefits and disadvantages right now to the Presup process, which I'll talk about in a minute. So it's mostly, it can be used for lots of things. It's anything you want to get feedback from the FDA. What we use it for is mostly protocol development, especially for chemistry, when you're trying to understand exactly what the FDA wants. Or if you're looking to justify out of testing and you think you have a good rationale, it's a good way to kind of communicate with the FDA about that rationale to get feedback. But there are a lot of reasons we do it for like toxicology, blood work that may be different, like getting out of a dog thrombo test, for example, things like this, we use a Presup for. So there's a lot of published internal FDA policies that they sometimes have to follow that we want to make sure we understand. There's a lot of things that we want to know why we're submitting a process for. When it's interesting, we submit FDA Presubs most of the time before testing, but I've seen companies use them anytime during the submission process to understand more about how this Presubs work or how the FDA may accept something. And we showed you how. Now before I want to talk about the current state of the Presup process. For a long time, we used it a lot and we loved it. We had great communication with the FDA. Everything they decided on, we were able to hold them to on the back end. Recently, we've been having some problems with the Presup, just to be completely frank. We've had a hard time scheduling them. The FDA's been very swamped, like all of us. And then a lot of times, the Presup ends up getting this cycle where the FDA wants us more stuff, and we go and get more stuff and do a new Presup, and they want more stuff. I'm at the company right now that we've been through 4 Presubs on the same question, okay? So I'm not gonna tell you guys never to run a Presub because I do think they're still useful. I just think you need to be careful on exactly why you're running a Presub. Some of my clients find it actually easier now just to do the testing and do the justifications and deal with them on the back end than trying to do a Presub up front. I'm still a fan of it. I love the concept of a Presub. I think we just have to make sure that the FDA is still like back to kind of where they were pre-COVID with the amount of work that they have and the amount of support that they have. Okay, so the last thing we're going to talk about today, and I got 5 minutes to go through this, so I'm going to try to do it fast, is the new guidance document on skin contacting material. I love this guidance document. It makes me see that the FDA is thinking about risk-based approach, right? Because they're saying, if your device contacts the skin, just don't test it every single time. Like, look at the risk and look at the materials and justify if it needs to be tested. So, what materials are included in these and what not? So this is what we call the NICE list. These are all the materials that the FDA considers safe enough that you shouldn't have to do testing on it. Now they put a lot of caveats in there as far as your processing, including sterilization and packaging. You still have to do all of that work. But these are the materials that the FDA feels confident enough for skin contacting devices to be safe, okay? This is a great step forward. If your materials are on this, it's easy. And we've used this right now, even though it's in draft and the FDA hates using it because it's in draft, we still reference it for our justifications because it's out there and it's a current thought process from the FDA. This is what we call the naughty list. These are the materials that are specifically not supposed to be like justified, that should be tested. Now a lot of these make sense to me, right? So if you look at fluids and creams, or you're looking in situ polymerization materials, breached or compromised surfaces, reprocessed single-use devices, like those kind of make sense. The one that I always have the hard time with is the first one, metals, okay? Metals. So basically what the FDA is saying is if you have a piece of stainless steel that contacts your body, on your skin, you have to test it. They're not really saying that. What they're saying is that you can't use this guidance document for it. You can still use the concepts, but what they're saying is that metals cannot be applied under this draft. I think, talking to them, I think their reasoning is because they're worried about nickel allergies. And so they're worried about specific metals that may have nickel in it, and they're worried about irritation or sensitization more specifically for nickel. But we're still kind of, we have comments out on this. We're talking with the FDA, trying to understand more. But this is, right now, the materials that are not included. So that's why we look at metals. We think about nickel sensitivity. And so do you have to test metal components? Like I said, no. You follow the same justification outlines as everything else, and maybe you can justify out of testing metals, just you can't use the guidance in this document. Okay, so I just want to let you guys know that with this draft document, we still use it, even though it's draft. I've heard the FDA come back to us and say, you can't reference that because it's draft, it's not official. I respond and say, I know it's draft, but the concepts are good, and I'm just letting you know that I agree with your concepts. So, and that's worked pretty well up to this point. So that's basically what a statement of the biocompatibility should be done in those, if you're using the draft guidance document. I also want to let you know that I flew through this, and I'm sorry. 30 minutes is not enough time for me to go over everything. If you are interested in more educational content, we are now going back to doing seminars. We have one in Chicago coming up. We also do have tons of content on our website, us and our sister company, Sterigenics. So please let us know if we can help with more. And at this time, thank you. I'll be out here if you have any questions. Please take anything you want, but thank you for taking your time. And give me your business cards if you want a copy of my presentation. So thank you.